name is Brendan Hardy, and today I will be discussing the processes of human-to-human transmission and viable detection methods for SARS-CoV-2. At the end of this presentation, I hope that you are first and foremost able to identify the SARS-CoV-2 genome and its primary components. This is important as, depending on the diagnostic test used, different genes or proteins of SARS-CoV-2 will be targeted. Next, a brief overview of human-to-human transmission of SARS-CoV-2 will occur in order to better understand how current diagnostic tests function and why these tests are so crucial in stopping the spread of coronavirus. Lastly, 6 widely implemented diagnostic assays will be assessed, including RNA amplification-based assays, isothermal amplification-based assays, next-generation sequencing, CRISPR technologies, serology, and antigen-based testing. A total of 7 electronic databases were utilized via EBSCOhost Web, some of which included HealthSource, Nursing Academic Edition, and Medline with full text. To best reflect the research question, the following search terms were implemented, and this yielded 4,325 search results. Of these results, only 20 articles were quantified as being relevant to the initial research question via pre-selected inclusion criteria, such as being recently published from 2020 to 2021. The global emergency of the COVID-19 pandemic has a ground zero that is located within the Wuhan province of China, originally emerging and spreading from that province in December of 2019. However, this is actually the third endemic spread of a more severe strain of human coronavirus. The originally identified severe acute respiratory syndrome coronavirus, or SARS-CoV, was first reported in November of 2002. Then, emerging in 2012, the Middle East respiratory syndrome coronavirus, or MERS-CoV, began infecting humans with a possible zoonotic source of dromedary camels. Regardless, all variants of human coronaviruses are positive sense, single-stranded RNA viruses that are known to infect both birds and mammals. There are 4 genera of coronaviruses within the family Coronaviridae, and these include alpha, beta, gamma, and delta. SARS-CoV, MERS-CoV, and SARS-CoV-2 are all included within the beta coronavirus genera. The genome structure of SARS-CoV-2 includes 14 non-structural open reading frames, or ORFs, and 4 structural proteins. These proteins are imperative for viral replication. The 4 structural proteins that are encoded includes the spike surface glycoproteins, membrane proteins, envelope proteins, and nucleocapsid proteins. Despite the severity of COVID-19 in some instances, 4 of the most common types of human coronaviruses identified as 229E, NL63, OC43, and HKU1 typically cause only mild to moderate upper respiratory tract illnesses. Specifically, HCOV, 229E, and HCOV, OC43 have been identified as being responsible for select cases of the common cold. Therefore, there is a wide range of human coronaviruses that all vary in severity of the symptoms that they cause. For clarification, the graph shown here displays the taxonomy of all coronaviruses, beginning with the Nidoviralis order, Coronaviridae family, and Orthocoronavirinae subfamily. From this subfamily, the 4 genera of coronaviruses are shown in detail, including beta coronaviruses, of which SARS-CoV-2 is identified as. You can note that most of these coronaviruses, including those within the gamma and delta coronavirus genera, are zoonotic in origin. Coronaviruses belonging to the gamma and delta genera primarily infect non-human mammals or birds. Again, for further clarification, the figure shown displays the viral genome structure of SARS-CoV-2. ORF1ab and RNA-dependent RNA polymerase, or RT-RPA, are non-structural proteins encoded within the 14 open reading frames of the genome. While non-structural, these genetic materials can be targeted for detection. In other words, depending on the diagnostic test, if ORF1ab or RT-RPA is detected, then the detection of SARS-CoV-2 infection is quantified. Otherwise, we can clearly see the 4 aforementioned structural proteins of the virus. Whereas the S protein is crucial for binding the virus to surfaces of host cells, the M protein promotes assembly of virus particles, the E protein enables the release of virions to increase transmissibility, and the N protein is involved in RNA replication of the virus. The single strand of nucleocapsid or N proteins in the middle of the genome, makes up the single-stranded RNA. Thus, each protein plays its role in attachment to host cells, viral assembly, RNA replication, and finally the release of further virions. Overall, viral transmission of SARS-CoV-2 has been almost universally determined to be through airborne contamination via aerosolized respiratory droplets or direct contact via self-inoculation from contaminated hands or surfaces. 2 studies notated that a viral suspension within aerosol remained viable for up to 3 hours after expulsion of respiratory droplets. While concerning surface transmission, the virus was shown to remain stable on plastic and stainless steel surfaces for up to 72 hours. Other surfaces such as copper and cardboard were more conflicting in their results. Following viral transmission, the median for symptom onset was roughly 5 days, with greater than 90% of individuals who develop symptoms doing so within 14 days following infection. Additionally, while SARS-CoV-2 maintains an overall lower mortality rate in comparison to SARS-CoV and MERS-CoV, it has been determined to have a higher degree of transmissibility. The reproductive number of SARS-CoV-2 has been variable in its expression from 2 to 5.7, meaning that, on average, 2 to 5.7 people become infected from one single person. Furthermore, while asymptomatic transmission does occur, it was determined that transmission appears to be 10 to 20 times more efficient following symptom onset. Concerning infection via viral entry into the host cell, this occurs by way of the spike surface glycoprotein and its 2 subunits, S1, which is the receptor binding domain, or RBD, and S2, which involves cell membrane fusion. Whereas S1 plays a vital role in viral entry, S2 facilitates fusion between the viral envelope and host cell membrane, allowing for host cell infection to occur. Pertaining to viral affinity, SARS-CoV-2 targets ciliated bronchial epithelial cells and type 2 pneumocytes with ACE2 receptors, thereby largely impacting the pulmonary system. Human ACE2 proteins are predominantly located within the lungs, kidneys, gastrointestinal tract, heart, liver, and blood vessels, which helps to explain why there is such a varying degree of symptoms in bodily systems impacted by infection. The most widely implemented procedures for SARS-CoV-2 detection are RNA amplification-based tests. These tests detect the presence of viral RNA by reverse transcription to separate the target viral RNA from the sample. Through the reverse transcriptase enzyme, viral RNA is converted to complementary DNA, which is then amplified at 3 different reaction temperatures. Fluorescent detection is used to convey detection of the amplified viral material, resulting in a positive test result. Of these tests, the RT-PCR method, which stands for Reverse Transcription Polymerase Chain Reaction, has been reported as the gold standard for SARS-CoV-2 diagnosis. However, gold standard remains in quotation, as there still exists a lack of a clear-cut gold standard given the novelty of this virus. As previously stated, genes or proteins of the viral genome are targeted for viral detection. RT-PCR was developed to target 2 to 3 genes, or one target gene is non-specific to detect the presence of coronavirus, and the other target gene is specific to SARS-CoV-2. This helps to reduce cross-reactivity with the other 6 known human coronaviruses. While the CDC recommends targeting 2 nucleocapsid proteins, N1 and N2, the World Health Organization recommends targeting the envelope protein followed by confirmation via RNA-dependent RNA polymerase, or RT-RPA. Despite widespread use, RT-PCR has been conveyed within the research as being complex due to its demand for highly experimental equipment and reagents, expensive, as well as time-consuming due to the logistical requirement of sample transport to qualified laboratories. This often extends turnaround time beyond a 24-hour period. Additionally, clinical sensitivity of RT-PCR methods decrease as the disease naturally progresses. This is due to a variable duration of viral shedding from case to case and further considerations of viral load. If viral load is low enough in situations such as asymptomatic cases, early infection, or later phases of infection, then there is a lower threshold for viral RNA detection to occur. SARS-CoV-2 viral load peaks within the first week following symptom onset, but the subsequent drop in viral load afterwards decreases the positivity rate. This has resulted in an estimated 30% false negativity rate for RT-PCR methods. In fact, positivity rate has been found to plummet to as low as 5.4% during the 5th week following symptom onset. Therefore, RT-PCR methods do not directly measure infectious virus, but rather detect the presence of viral RNA extracted from the collected sample. RT-PCR assays do not accurately measure the capacity for transmission to occur. Much like traditional RT-PCR, isothermal amplification-based methods also detect the presence of viral RNA via reverse transcription, followed by amplification of the resulting complementary DNA. However, the distinction between these two assays is made via isothermal conditions, with no temperature change required throughout the amplification process. Furthermore, in comparison to the 2 to 3 genes targeted in RT-PCR, isothermal amplification-based techniques recognize 4 to 6 distinct gene sequences of the SARS-CoV-2 genome. As a result, test turnaround time is more rapid, more clinically sensitive, and more clinically specific in comparison to RT-PCR. These assays also do not require expensive laboratory equipment or instruments to perform. 2 of the most documented isothermal amplification-based tests include reverse transcription loop-mediated isothermal amplification, or RT-LAMP, and reverse transcription recombinase polymerase amplification, or RT-RPA. Isothermal amplification-based tests are acknowledged for their viable point-of-care capabilities. Studies have indicated that results from RT-LAMP become available within less than 1 hour following test completion, which is significant while comparing it to the multiple hours to days that it could take to obtain conventional RT-PCR results. Test turnaround time for RT-RPA has been even more promising, being reported at less than 20 minutes from test completion. Additionally, detection time could be further expedited by concomitant application of either RT-LAMP or RT-RPA with CRISPR diagnostics. In other words, combining isothermal techniques with CRISPR has shown to improve the point-of-care capabilities of otherwise employing each method alone. Next-generation sequencing employs an unbiased mechanism to reveal the genomic sequence of novel viral pathogens, even those that have yet to be identified. And being unbiased, NGS requires no prior knowledge of the virus that it attempts to sequence. Genomic sequencing was utilized to separate and identify the SARS-CoV-2 genome, which was initially designated 2019 nCoV from BAL sampling of patients in Wuhan. These early reports revealed a genomic sequence homology between these collected BAL samples of 99.9%, thereby confirming the presence of the single novel pathogen that had been inducing severe pneumonia. Apart from wholly identifying a new strain of human coronavirus, NGS was also the initial step in producing the necessary diagnostic primers used in RT-PCR, as well as directing the development of antiviral therapies and vaccines. However, limitations to this technology has included overall complexities with protocol or equipment, general expense, and long turnaround times of 24 to 48 hours for results. Therefore, this diagnostic test is simply not practical for widespread implementation. CRISPR, otherwise known as clustered, regularly interspaced, short palindromic repeats, is renowned as an adaptive genome editing toolkit that has implications for both diagnostics and therapeutics. The CRISPR-Cas system involves the use of Cas proteins, otherwise known as effector enzymes. It is important to note that different CRISPR-based tests will utilize different Cas proteins to induce this enzymatic activity. These Cas enzymes are used to recognize specific sequences of the SARS-CoV-2 genome. Guide RNA, which is complementary to these target sequences, work to guide Cas enzymes to their targets for binding to occur. After binding with these genome sequences, enzymatic activity degrades all nearby RNA, a process that is known as collateral cleavage. Fluorescent reporter molecules that have been added to the reaction are also cut via this collateral cleavage, which results in a fluorescent signal release. This release confirms the presence of viral genetic material. Because of reduced turnaround times, all CRISPR-based assays offer a viable point-of-care method for diagnosis that could be employed within the field. This is due to its overall simplicity, ease of use, portability, and cost-effectiveness. In fact, it has been estimated that a single paper-based SHERLOCK test was valued as low as 61 cents in U.S. currency. As previously stated, different CRISPR systems employ the use of different Cas enzymes. For instance, DETECTR allows for Cas12a detection of SARS-CoV-2 genomic sequences following RT-LAMP protocol. In DETECTR, after RT-LAMP, Cas12a specifically targets the envelope and nucleocapsid proteins of the SARS-CoV-2 genome. Following collateral cleavage and the subsequent cutting of reporter molecules, results are displayed via lateral flow visualization. DETECTR has been shown to provide rapid results within less than 40 minutes and has also been shown to maintain a clinical sensitivity and specificity of 95% and 100% respectively. Additionally, it has been reported that Cas12-based detection systems maintain a higher sensitivity and time effectiveness with regards to turnaround time in comparison to Cas13-based assays. SHERLOCK was the first FDA-approved CRISPR-Cas system on the market for clinical diagnosis of viruses and has been previously used for detection of both Zika and Dengue viruses. It utilizes Cas13a detection of SARS-CoV-2 genomic sequences following isothermal amplification of the sample via RT-RPA protocol. Cas13a is applied to target the spike surface glycoprotein and ORF1ab. Like DETECTR, SHERLOCK provides for a rapid turnaround time with results within 1 hour following collateral cleavage and subsequent cutting of reporter RNA. Unique to SHERLOCK, however, is the fact that it can detect the presence of viral genetic material even if low concentrations of it exist within the collected sample. This negates the limitation of traditional RT-PCR, where decreased test sensitivity is often observed in samples with low viral load. Lastly, CONAN employs the use of the Cas3 enzyme followed by RT-LAMP protocol, which is similar to DETECTR. However, CONAN specifically targets two nucleocapsid proteins, N1 and N2. CONAN offers a rapid and sensitive viral detection system. Despite this, DETECTR and SHERLOCK are most widely implemented for clinical detection of COVID-19. Serological immunoassays are largely employed for epidemiological purposes to examine community prevalence of infection. These diagnostic tests, such as ELISA or lateral flow immunoassays, are designed to detect the humoral development of antibodies, or immunoglobulins, in response to antigens, otherwise known as viral proteins. In fact, ELISA and lateral flow immunoassays are the most widely implemented techniques for antibody detection against SARS-CoV-2 antigens. The three major antibody isotypes that have been correlated with SARS-CoV-2 infection include IgA, IgM, and IgG. Both IgA and IgM were associated with early phases of infection, being detectable within collected samples at a median of 5 days post-symptom onset. Despite this, in two separate studies, IgA maintained a higher positivity rate during acute phases of infection in comparison to IgM. IgG was universally correlated with late phases of infection, and thus better reflects long-term immune response. IgG was detected at a median of 14 days post-symptom onset. Overall, an increased viral load within collected samples has resulted in a proportional increase in antibody titers, so antibody levels appeared to correlate well with severity of infection. Serological exams against SARS-CoV-2 primarily detect immunoglobulin response to two antigenic targets, the nucleocapsid and spike proteins, more specifically the S1 subunit of the spike protein. While it has been reported that targeting N viral proteins increases test sensitivity in comparison to S viral proteins during early infection, N viral proteins appear to be more of a risk factor for cross-reactivity with other endemic human coronaviruses. For instance, N protein homology between SARS-CoV and SARS-CoV-2 was reported to be as high as 90% in one study, whereas S protein homology was 77%, and S1 subunit homology was reported at an even lower 66%. Thus, the S1 subunit could present a more specific target antigens for SARS-CoV-2 detection. Other limitations include that the degree of antibody protection against COVID-19 infection is largely dependent upon age, gender, nutritional status, comorbidities, medications, and severity of symptoms. As serological tests measure humoral response to infection, which can take several days or weeks to develop, these assays are also not a reliable measurement of current infection, but rather infection that has occurred in the past. Adding to this, immunoassays are not as clinically specific as RNA amplification-based assays, such as RT-PCR. However, concomitant use of RT-PCR with serology has been shown to increase detection capacity. A combination of RT-PCR with ELISA was shown to increase detection rate by 98.5% within 2 studies. This chart does an excellent job differentiating between the measurements made by nucleic acid amplification tests, such as conventional RT-PCR, and antibody detection tests, such as ELISA. While these diagnostic tests both work well to facilitate contact tracing, especially when utilized together for epidemiological purposes, one is utilized to measure current or recent infection, whereas the other can only detect past exposure to COVID-19 infection. The distinction between these two concepts is significant. In having a mechanism of action that is antagonistic to antibody detection methods, antigen tests detect viral proteins via capture reagents, also known as antibodies. The Sofia 2 SARS antigen FIA was the first antigen-based test to receive an FDA-issued emergency use authorization with results within 15 minutes. Like serological exams, these are inexpensive and simple-to-use tests with real point-of-care capabilities, as they are often termed rapid diagnostic tests, or RDTs. Most antigen tests are handheld, portable assays designed for rapid detection of viral RNA from nasopharyngeal specimens. However, these tests have also been found to retain less sensitivity and specificity in comparison to other diagnostic tests, such as RT-PCR. Moreover, available data regarding the 4 commercially available antigen-based rapid diagnostic tests have indicated extreme variability in their clinical sensitivity, ranging from 16.7% to 85%. Thus, while rapid, these assays may not be the most reliable method for controlling the spread of COVID-19. In summary, the viability of employing each method individually during the ongoing phases of vaccine distribution remains unclear. However, concomitant diagnostic practices have shown promise in reducing the limitations that may result from individual use of one of these tests alone. For instance, DETECTR, with its combined implementation of the Cas12a enzyme and isothermal amplification, has improved clinical sensitivity and specificity as a result. Furthermore, DETECTR remains a viable point-of-care detection method. For epidemiological purposes, multiplexed immunoassays should be considered to monitor the prevalence of SARS-CoV-2 infection within communities as vaccines continue to be distributed. Ultimately, however, future research is required to confirm the efficacy of novel diagnostic methods in the event of prospective viral outbreaks. This is especially true while regarding more novel strains of SARS-CoV-2 that are appearing internationally. What follows are all of my references that were utilized for this literature review. Thank you so much for your time.